here. There's no agriculture there. There's essentially no people there. I should back up. My color scheme here is red is high abundance of algae and blue is low. Okay, so we have this big bloom of cyanobacteria right here in north central Florida Bay, out in the middle of nowhere. Look up here, Biscayne Bay. Using the color scheme I've got here, this shows really low abundance of algae in Biscayne Bay, right next to an urban area with 2.5 million people. So that tells you there's something odd going on here. Okay. And so after I started studying the cyanobacteria bloom in Florida Bay, then I started looking at the linkages with everything else. So over the years, I've been sampling all throughout. I've been studying blooms in Biscayne Bay, the red tidal on the west coast, and the Lake Okeechobee, the Clusatch River, St. Lucie, and so on. So I'll be showing you some of that today. Okay, so I'm going to first try to figure out what's going on with this bloom of cyanobacteria in Florida Bay. And I quickly learned something very odd's going on, because remember, with sewage or farm runoff, you're going to have nitrogen and phosphorus together in the same location in about that 16-to-1 ratio. If you look at Florida Bay, you see high phosphorus, and this pointer's not showing up too well, you have high phosphorus in the western part of the bay, and high nitrogen in the eastern part of the bay. That says there's got to be two different sources. They're not coming from the same source. And again, I've just plotted up the chemical data here. Blue dots indicate a high nitrogen to phosphorus ratio. There's excess nitrogen. What the plants are going to run out of first is phosphorus. It's called a phosphorus-limited ecosystem. And then the red dots over here indicate the opposite, a low nitrogen-phosphorus ratio. So it's excess phosphorus, and you're going to run out of nitrogen first. That 16-to-1 ratio, which is sort of optimum for the algae, is right there in the middle where you get the algal bloom. There's the algae, you don't get a big algal bloom if you have a lot of phosphorus but no nitrogen, or vice versa. You got to have both of those nutrients. Here we actually do what we call nutrient bioassays. The previous data was the chemical data, here's the biological data confirming that, yep, these algae are in fact phosphorus limited over here in the east and nitrogen limited in the west. Okay, so you have phosphorus in the west, nitrogen in the east. Where they meet in the middle in that 16-to-1 ratio is where you get that bloom. Okay, so where is that phosphorus coming from? Turns out that's primarily a natural source. I won't go into all the gory details of this. It took a long time to figure this out, but basically, some of you know, we have big phosphate deposits up there east of Tampa Bay. It's a huge mining operation up there. And you can, when I looked at some unpublished geological data, I found out that you've got these deposits going underground all the way down to western Florida Bay. It also intersects up here in the 10,000 islands there. And that's exactly where you see high phosphorus, here in the western Florida Bay and up there in the 10,000 islands area. So we just expand this now. Bottom line, you look at these ratios. Eastern Florida Bay, I don't have the data shown here, but Biscayne Bay is also phosphorus limited, just as the Everglades up in here are also phosphorus limited. Once you get over to western Florida Bay and all the way up the west coast, it's a nitrogen limited ecosystem. You've got excess phosphorus from those natural phosphorus deposits. Nitrogen is what's going to cause you problems. And that's very important because the government officials often just say nutrients and they don't distinguish between them. And that's just nutrient bioassay data showing the same thing. The entire west coast is nitrogen limited. So we know where the phosphorus is coming from. Now where's this nitrogen coming from? Let me back up. That phosphorus has been there for millions of years, okay? But you didn't have blooms all that time because there was no nitrogen, okay? So where's this nitrogen coming from? Well, this yellow shows fresh water coming out of the Everglades. Matches where the nitrogen is. So it tells us the nitrogen is coming from the Everglades. So we just look at the drainage system. Start up here in Lake Okeechobee. The Everglades agricultural area, that's the northern third of the Everglades, which has been turned into agriculture, which is about 90% sugarcane. And then it flows down through this canal system all the way into eastern Florida Bay. So we're going to do a transect from the sugarcane fields down through the Everglades and down into Florida Bay. Here's the sugarcane fields over here. Here's Florida Bay over here. Red is concentrations of phosphorus. So you see phosphorus coming out of the sugarcane fields, and it eventually gets used up by the plants because the Everglades are phosphorus limited. So the phosphorus never makes it down into eastern Florida Bay, which is what you saw on the map. That phosphorus never makes it down there. But the fact that the phosphorus does make it into the Everglades here is what led to the federal lawsuit back in 1988, which then led to the whole Comprehensive Everglades Restoration Plan. What that plan totally ignored was the nitrogen. They still continue to ignore the nitrogen half of the story. Well, the nitrogen here is the blue line. And I've adjusted the scales. Here's the nitrogen scale over here and the phosphorus scale over here, such that if those nutrients were in that 16-to-1 ratio, those two lines would be on top of each other. So what that tells you is there's a huge excess of nitrogen. The N to P ratio is like 200-to-1, way over the 16-to-1 that the plants need. So initially, you see the nitrogen being used up as the water flows south, being used up by the plants. But once the plants run out of phosphorus, they don't really have much use for the nitrogen anymore. And so the rest of that nitrogen just gets flushed all the way on down into Florida Bay. That's the source of that high nitrogen you see in eastern Florida Bay. Now, this phosphorus is primarily fertilizer, and you might think that nitrogen as well. But if you think about it, why would a farmer add fertilizer in a 200-to-1 ratio when you only need 16-to-1? If that's fertilizer, then you're flushing most of your fertilizer all the way down into Florida Bay. Turns out that's also primarily natural nitrogen. I need to explain that now. If you think about the Everglades, why you even have it in Everglades is when you think of Florida as being very flat, but in fact, for geological reasons I won't go into here, you've got a ridge right here, it's called the Atlantic Coastal Ridge, and another ridge right down the middle of the state, and so you've got a trough in the middle. So if you look at it from the side, you've got a ridge over here, a trough in the middle. So as Florida came out of the last ice age, sea level's rising and it's becoming more humid, you start filling this with water. That's how you build a swamp. Okay? You get your wetlands, and as you build this up, all the dead vegetation goes down underwater, becomes anaerobic. You build up organic peat. This is what wetlands do. They absorb the nutrients and store it in organic peat. So over the course of 5,000 years, which is about how old the Everglades are, you've completely filled up this trough with organic peat up to the surface. As long as it's a wetland and that organic peat is underwater, you preserve those nutrients. And basically, the wetlands act as a nutrient sink. So only clean water is coming out of the wetlands. But now humans come along and now want to drain the wetland to use it for agriculture or urban areas and whatever. You expose that organic peat to the air, and now bacteria break it down. Okay? And basically, this leads to what we call land subsidence. That organic peat is like 99% organic material. You decompose it, it just goes into CO2. And all your land soil disappears. So here's an example. 1924, the surface of the land here in your Bell Glade was right there. And when this picture was taken, I'm not sure exactly when, like the 1990s or something, the surface of the land is there. You've lost that much organic peat. And in doing so, you released all that nitrogen downstream. Another picture. Here's a home that was built in 1930s out in the Everglades. Initially, that was the surface of the soil. And now, during that period of time, you've lost all that organic peat. And you've lost all the nitrogen that was in that organic peat. So what's happened, basically, is 5,000 years worth of nitrogen has been released in the past 50 years and gone down into eastern Florida Bay. And basically, as this land subsides, then they have to keep on adding more steps every so often so they can get up to the front door. Okay, so let's look at the sugarcane history in the northern third of the Everglades. It just sort of putters along in the first half of the 20th century. 1959, it suddenly shoots up. What changed in 1959? Well, that's the Cuban Revolution. Okay, United States, we're in the middle of the Cold War. United States happy with having a communist dictator only 100 miles to the south, and so they tried various ways of trying to get rid of Fidel Castro. The Bay of Pigs didn't work out too well, the exploding cigars didn't work out too well, etc. So they decided to try to destroy the Cuban economy, which at that time was primarily sugarcane. So it's a complicated story, but basically you've got this complex series of laws that subsidize the sugarcane industry. And that's what promoted the sudden increase in sugarcane growth in the northern third of the Everglades. And this was so successful getting rid of Fidel Castro that we've continued, and we continue today, to subsidize the sugarcane industry to the tune of maybe $2 to $4 billion a year. Okay, well that was in the 60s, but we didn't see the algal blooms in Florida Bay, we didn't see the ecological problems in the Everglades until the 1980s. Why is that? Because initially, in order to drain all this land here, they reversed the flow of water in South Florida. They actually installed giant pumps and pumped all this water back upstream into Lake Okeechobee, and then down the Clusatch River to the west and St. Lucie Canal to the east, in order to keep the sugarcane fields dry during the wet season. I'll show you some data here. At this point here, halfway between the sugarcane fields and Florida Bay down here. What you see here is very low, this is flow rates heading south. You see very low flow, you have a few hurricanes and so on, but basically low flow until the 1980s. What happened was, in the 60s and 70s, you see huge amounts of nutrients going into Lake Okeechobee from the sugarcane fields, and then huge algal blooms, fish kills, and so on, and then lawsuits. So after that huge disaster, they decided to reverse the flow of water again. So in the 1980s, they start sending the water south. And that's what you see here, 1980s. And not soon after, you start seeing huge ecological changes in the Everglades, which led to the lawsuit, and then algal blooms in Florida Bay. This shows water flow into Florida Bay. Again, low levels before 1980, and then after 1980, you see this large increase. And we don't have data on those algal blooms back then, but the fishermen are out there every day, and they could see what was going on. From 1981 onward, the water started getting cloudy, having a hard time seeing the bottom. Just all these things say the same thing. Commercial fishermen saw the water first by becoming dirty, then algae bloom started, and then seagrass died off. Total change of the ecosystem. And well, I'll just skip over that one. Just show you again, the blue is high salinity water, and orange is low salinity water. That indicates fresh water coming in. So during a drought, you have very little fresh water in the bay. It's very salty. Then you come out of the drought, and now you have a lot of fresh water coming out from the Everglades and into the bay. Now we look at the nitrogen. During a drought, when there's very little fresh water coming in, very little nitrogen. Then coming out of the drought, you pump lots of fresh water in from the Everglades down into the eastern Florida Bay. You flood the bay with nitrogen. When that nitrogen now meets with the phosphorus over here in the west, that's where you get the bloom. Right there in the middle. So I'm going to take some data at those 4 stations right there. And here's 15 years worth of data. Blue is the amount of water flow into the bay. We have a very distinct wet season, dry season cycle. So you see 15 years, wet season, dry season, up and down. And then green is the size of the algal blooms. Basically shows the same thing. If we just take monthly averages, here, blue is water flow into the bay. Green is the size of those algal blooms. Here's some dry years and here's some wet years. You can see how they correlate. That's specifically cyanobacteria. It just tells you that, in fact, those blooms are primarily cyanobacteria. You can also look at annual averages because you have wet years and dry years because of El Nino and other long-term climate shifts. And again, here's some wet years and here's some dry years. Okay, well, the state government and the sugarcane farmers were not too happy with me showing these data, so they got some people to take the data set and concluded there in fact is no correlation whatsoever between water flow runoff into the bay from the sugarcane fields and the algal blooms. Well, you can look at the data and decide for yourself. There seems to be a correlation that every time you dump more fresh water in the bay from the sugarcane farms, you get larger algal blooms. And they were using the exact same data set I was. How can we come to such different conclusions? Well, the reason is because neither of us used the full data set. What I did is I took the data at those 4 stations right there in red, and I showed there is a correlation. These other folks took the data in blue. In other words, I took data where the blooms are and showed there is a correlation between runoff and the algal blooms. They selected out just the data where there is no bloom and concluded there's no correlation. Well, of course there's no correlation because there's no bloom. Okay? Point is, you really have to look at the details, the fine print of these kind of publications to see what's going on. Technically, they did not lie. There was no correlation, but they cherry-picked the data to make sure they got what the state government and the sugarcane growers wanted. Okay? So you always have to be careful when you hear people saying certain things. They're not necessarily lying, but they certainly can manipulate things and be very, very misleading. And just very quickly, I'm not going to go into details, it's more complex, but this dirty water does make it out of Florida Bay, down, out over the coral reefs. Down through the passes here, it gets picked up by the Gulf Stream, carried out over here, and so on. Satellite imagery. And indeed, if you look, this is one particular reef, the blue is the amount of this nitrogen-rich water coming out of the Everglades, out over the coral reefs, and then green is the abundance of algae on the coral reefs. You see this long-term correlation. And indeed, here's some pictures. Here's a large coral head down in the Keys, hundreds of years old, been growing perfectly fine for hundreds of years. This picture is taken like in the 80s or something. And then 10 years later, come back completely dead, overgrown with algae. Here's another one, coral head 1994, doing just fine for hundreds of years. Come back 6 years later, completely dead, completely overgrown with algae. A colleague of mine was studying one particular reef for other reasons. Back in 1975, over 60% of the coral reefs were covered with living coral. 1983, it's dropped a little bit. Comes back in the 90s. Now remember, this is a time period in which now you're dumping all this nitrogen-rich water down into Florida Bay. By 1996, you're down to only a little over 10% living coral cover. And by this point, only like 5 or 6%. Okay. Another colleague of mine then seen what was going on. He didn't get going until 1996, but looking at the entire Florida Keys, 160 stations, 1996, 10% living coral, just 3 years later, down to 6%. Now, there's a lot of other things going on. Corals around the world are dying for a number of different reasons. Global warming, acidification, so on. So I can't ascribe all this decline to the nutrients, but we do know high nutrients can lead to algae overgrowing the reefs and killing them, so that's probably at least a factor anyway. Okay, now let's move up north to the algal blooms that you've been hearing about in the news. Okay, so remember for many decades, this nutrient-rich water coming out of sugarcane fields was back pumped in Lake Okeechobee. Now, the Clusatch River was not initially connected to the lake. It was a separate watershed. But in order to drain the Everglades and sugarcane fields, you had to pump water back up into that lake. You have to get that water out someplace. So they connected the lake to the river, so you can now dump water from the lake down the Clusatch River. Here's some data. Here's Lake Okeechobee, here's the Clusatch River. These red dots just simply mean there's a high abundance of these cyanobacteria in the river. Here's a picture, here's one of the dams on the river. You can see this dense bloom of cyanobacteria on that river. Every time I do a cruise along the west coast of Florida, you see higher abundance of algae right there near the mouth of the Clusatch River. Here's the Clusatch River, here's Sanibel Island, Fort Myers. So clearly the Clusatch River is a major source of nutrients to the west coast. Okay, here it is again. Basically, Estero Bay, Clusatch Estuary, Pine Island Sound. This is during the dry season when there's very little river flow. You see low abundance of algae. During the wet season, you have high abundance of algae. Here's 5 years worth of data. Ignore the bottom half, just look at the top half. 5 years worth of data. Blue is river flow, wet season, dry season, wet season, dry season. And the green is the size of the algal blooms in Charlotte Harbor. There's Pine Island Sound, same thing. And there's Estero Bay. Very good correlation. Now, up to now, I've just been saying algae. I can tell you what type of algae it is, okay? Now I want to talk about one particular species of algae. This one here. This is a dinoflagellate called Crynia brevis. This is the one that causes the Florida red tide. And it produces this toxin called brevetoxin. Okay? And it leads to massive fish kills. Here's a harbor completely filled with dead, rotting fish. Kills manatees, dolphins, sea turtles, and so on. You've seen all this in the news. Okay, what I've taken here is 50 years worth of data and just plotted up, see on average, where do you tend to get the red tide? This is the hot spot, basically along the coastline from around Tampa Bay down to Naples. Okay, if you just plot, just do transects from the shoreline over here, going offshore, blue is salinity. So lower salinity as you move inshore, because that's where your fresh waters come in, your land runoff. And of course that's associated with nutrients. And then red is the average abundance of red tide. So clearly there's a lot more red tide inshore where you have your fresher water and more runoff of nutrient-rich land runoff. Okay. Here's the same plot, except now the blue is what the average red tide abundance looked like 50 years ago. Red is what it looks like now. And as on average, red tide is 15 times more abundant than it was 50 years ago. Now, red tide does occur naturally. We've got records to indicate, or certainly strongly suggest, that you did have red tide even as much as 500 years ago when the Spanish explorers first went along this coastline. Okay? But it's 15 times more abundant now than it used to be. I can't think of any natural sources of nutrients that would have increased 15-fold. What obviously has increased in South Florida over the last 50 years is the number of people and associated agriculture, etc. And so any area you take, you see blue is 50 years ago, and then here's today. You see this dramatic increase in the abundance of red tide. Just look right along the shoreline. That's the area that affects people the most, this dramatic increase. Sarasota Bay, Lemon Bay, all these places. Okay? If you just look at the densest blooms, how dense do they get? 50 years ago, it was like 2.5 million cells per liter. Now it's more like 35 million cells per liter. Okay? Again, like about a 15-fold increase. Well, once again, the state of Florida didn't like that I was saying that this red tide's not just natural, but rather than nutrient-rich runoff is making it worse. So they hired some people to come up with a counter report and concluded, nope, red tide's not increased at all. But once again, if you look at the fine print, what they really did, they didn't actually use the raw data like I did. They manipulated the data first. They categorized all the data. In other words, if the abundance of red tide was less than 1,000, then you call it category 1, etc. Anything over 100,000 is category 4. And notice these people, they did put in fine print that yes, that these numbers were chosen by the government officials. Okay. Well, basically, remember, category 4 is anything over 100,000. In other words, we've gone from category 4 to category 4. There's not been an increase in red tide. Okay? If you manipulate the data first in the right way, you can get any result you want. Okay, so you have to look at the fine print to see what people are doing. Okay, I mentioned there was no initial connection between Clusatch River and the lake. So the blue is the original watershed of the Clusatch River. But by now connecting it to the lake, and then back pumping water from the sugarcane fields back up into the lake, that's now part of the watershed. Similarly, the Kissimmee River Basin used to be a large area of wetlands as well. A lot of that was drained for now you have cattle ranches, dairy farms, citrus, and so on. So once again, you've taken that entire red area, you've taken wetlands that naturally are a nutrient sink, drained them, turned it into a nutrient source, and added in agriculture as even more nutrients. So the watershed is, first of all, 4 times larger than the natural watershed, but now you've got this huge excess of nutrients as well. So that's why the Clusatch River is such a major source of nutrients to the west coast now. So again, there's this dramatic increase in the sugarcane fields, the latter half of the 20th century. You can look at the long-term history of nutrients in ecosystems by taking what we call sediment cores, because of the way sediments accumulate over time. So here's some sediment cores, and you can age it. So this is what the nutrients looked like back in the year 1800, and this is what they look like now. And so you see this dramatic increase in all these nutrients just over the last few decades. And now we see the result with all these algal blooms throughout South Florida. It just shows the abundance of algae in this core as well. Okay, let's look at it from a seasonal point of view. From January through December, blue is rainfall, so here's our dry season, wet season, dry season. Orange is flow down the Clusatch River. And then red is average abundance of red tide. So it's a bit of a delay, but basically you can see, because it takes time for the algae to grow, but basically you can see once you dump all this fresh water downstream into the Gulf, you see an increase in average abundance of red tide. I'm going to show you 3 sequences real quick. This is in 2001. So here's a flow down the Clusatch River this year. This is the wet season here. That's the Peace River as well. Now, ignore the left-hand side, just look at the right-hand side. What we have here is black dots indicate a sample was taken and there's no red tide. The larger the red dot, the higher abundance of red tide. And the thickness of the blue line on the rivers here tells you how much water is coming down the river. So we'll start out in January, the dry season. So we're just go month by month. Dry season, nothing's happening. Now we have the wet season, starting to get river flow, and now you see the red tide developing and the coastal currents carrying it to the north there. And eventually it dwindles away. Let's take another sequence. This is in 2006. Start out in the dry season. You see now the beginning of the wet season, a little bit of red tide right there in the mouth of the Clusatch River, and then it expands as you move through the wet season. We'll take a third one, and this is 2004, when 4 hurricanes hit Florida. So you have a massive amount of rainfall dumped on the land, huge amount of runoff. Start out the dry season before the hurricanes. Now, I think Hurricane Charlie had just hit, so you're starting to get a lot of runoff down the rivers. September, no samples are even taken. Either the boats have been sunk or everyone's afraid to go out in the Gulf to collect any samples. Okay, now October, massive amounts of water coming out, but very little red tide down here. And now it's way down here. And then even further down. What's going on? Well, the red tide is a marine species. It doesn't like fresh water. What's actually happening, you have so much water coming down the Clusatch River. You have all this really dirty water coming. You see the color here. Massive plume of fresh water coming out of the river. The red tide it likes the nutrients in that water. It doesn't like the low salinity. So what you find is the red tide's living on just the front plume here, where this water is salty enough for the species. It gets the right mixture of salt and nutrients. A colleague of mine then took the data we have when we see the red tide way down here, and she could sort of do reverse modeling of the water flow, track it back, and sure enough, this water here where the red tide is came from the mouth of the Clusatch River. Okay, health effects. Red tide was initially known as neurotoxic shellfish poisoning because the shellfish, they filter the algae out of the water, use it for food, but they can't get rid of this toxin. It's a lipid-soluble toxin that accumulates in their tissues. It doesn't affect the shellfish, doesn't kill them or anything, but a human comes along and eats those shellfish, you end up very, very sick and in the hospital for long periods of time. But that is very rare now because we monitor for red tide, and when you have red tide, they shut down the shellfish harvesting. So that's very rare now. But if you look at hospital records on the West Coast, during times of red tide, you'll see a 40% increase in people going to the hospital for various gastrointestinal disorders. I'll come back to that in just a minute. The main thing that you see now, people are aware of, is the aerosol. This is a rather interesting toxin that gets up into the air and ends up like a tear gas. It irritates your eyes, nose, throat, lungs, and so on. And again, if you look at the hospital records, during times of red tide, you see about a 50% increase of people going to the hospital for various types of respiratory distress. So it's very clear it's having a huge impact on many people's health there on the West Coast. And of course now they're on the East Coast and up in the Panhandle area. Okay, this is probably the most complicated slide I'll show you. But basically what I've done is this concentration frequency spectrum. All we're doing is like, what percentage of the time do you take a sample and not find any red tide at all? Blue is what it was like 50 years ago. Red is what it is now. So the fraction of time you find no red tide has gone down. Same thing up to 1,000 cells per liter. That's considered background concentrations of the red tide. But for all these higher concentrations, you see higher fraction of time do you find these high concentrations of red tide than you had 50 years ago. Now what people focus on is anything over about 100,000 cells per liter. It's at that concentration that you can actually see discoloration in the water. You actually start getting the irritation from this tear gas type of toxin, and you start to see dead fish. That's your toxic levels. And that's what people focus on, anything over 100,000 cells per liter. And people know, you see these dead fish, they know not to eat it. But notice, people have always sort of ignored these sublethal concentrations. It's not, the toxin's there, it's just not enough to kill the fish. And I think what's happening, people think, well, the fish is alive, it must be okay, and they're eating these fish. I think that's why we're seeing a 40% increase people going to the hospital for gastrointestinal disorders. And indeed, we've found dolphins out in the Gulf that have died, even though there's no red tide around. It hasn't been for a long time, but you do the autopsy, they're packed full of this brevetoxin. What they're doing, I think, is they're eating the fish that have these sublethal concentrations. I'm not sure why the pointer's not. Okay. Right in here. These fish are exposed to these sublethal concentrations, so it's the toxins in the fish is not enough to kill them. It's just like you eating, say, swordfish that's got sublethal concentrations of methyl mercury in it. That's why you don't want to eat much swordfish or some of these other fish. If you eat just one fish a month, that's probably okay. If you start eating a lot of swordfish, you're going to come down with mercury poisoning at some point. Okay, what's this data showing that indeed you go out there during times no red tide around? I know you can't see the numbers, but basically this documents all these species of fish. You can find brevetoxin in the tissue. It's not enough to kill them, but it's there. So if you go out and eat those fish out there in the Gulf of Mexico, you're probably getting some brevetoxin in your diet. Okay, now let's go back to Florida Bay. Remember I said those are cyanobacteria. Lake Okeechobee, this is from a satellite image. Big blooms of cyanobacteria over this 800 square mile lake. That's a surface view. Clisette River, that's cyanobacteria. This is from 2 years ago, San Luis Estuary, famous guacamole blooms, and cyanobacteria. That's what it looks like when it starts dying. It turns that blue color. Well, we know cyanobacteria produce a number of toxins. The one that's best known is microcystin. Okay? And here's just some data I collected up in the lakes up in central Florida. This is abundance of algae. The larger the circle, the higher abundance of algae. So a lot of those lakes, again, excess nutrients, you're getting these blooms of cyanobacteria in those lakes. That shows that those algae are primarily cyanobacteria. And that's concentrations of this toxin, microcystin. Okay, so microcystin was actually discovered like 100 years ago over in Australia. A bunch of sheep died, and about the same time and location. So people looked into it, and it turns out they'd all been drinking from this pond over here. And that pond has got a big bloom of cyanobacteria. So when you look into it, you discover this toxin, microcystin. And since then we know a lot of animals have died, humans have died, and you also get short-term gastrointestinal disorders. Okay? So that's how you discover this toxin, because you get immediate effects. So a bunch of humans or animals get affected at the same time very quickly, and you can track it back. But once people started looking into what microcystin actually does to you at a biochemical level, we discover it can also lead to long-term liver damage, it's a tumor promoter, and can lead to things like liver cancer. Now think for a second, suppose this toxin only led to cancer or something like that. Cancer takes a long time to develop. If those sheep in Australia had only come down with cancer and not the other things, that'd been 10 or 20 years later. Well, who knows where those sheep would be, right? I mean, you would never have been able to track it back if there was no short-term effects of the toxin and only these long-term effects. So let's think about that. There's various compounds produced by cyanobacteria. Some just smell bad or make the water taste bad. Some are, if you go swimming in the water, all of a sudden your skin's on fire, severe skin irritation. Some can form short-term gastrointestinal disorders. I know it sounds kind of odd, but those I consider sort of the good toxins, because you know right away you're being exposed to it and you can do something about it. What happens if you have long-term neurological problems? Oops. Or lead to liver damage, or neurodegenerative diseases, or cancer. Those toxins do not have short-term effects, but you come down with a problem 10 or 20 years later. It's going to be very hard to discover that type of toxin if you only get the health effects 10 or 20 years later. Well, by accident, we think we've discovered one. It's called BMAA. And it turns out it's produced by, I know you can't see the numbers, but basically it's produced by essentially all cyanobacteria, as far as we can tell. And we now have good evidence it can lead to neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS. Now these are what we call the tangle diseases. The characteristic of these is that you get these protein tangles in your brain cells. And the main point here is they're only like 5% genetic. There's got to be some environmental stimulus or cause. We know some genes are involved, but it's got to be some kind of environmental factor playing the major role here. The exception to that is being Huntington's disease here. It's 100% genetic. We know what the gene mutation is for that. These others, there's got to be some kind of environmental cause. And there's lots of research been done on this for many decades, trying to figure out what these environmental causes may be. Again, characteristic of these protein tangles that accumulate in your brain until it kills off your brain cells. Well, the way this got discovered is a very unusual event back during World War II. As Americans and Japanese are fighting over the islands of the Western Pacific, including Guam, and because of the fighting, the native Chamorro people of Guam had to totally change their diet, to get enough food to survive and so on. At the same time, they had access to guns for the first time. As a result, they started shooting the fruit bats on the island of Guam for food. And they essentially, you can see this high abundance of fruit bats on the island of Guam, and they essentially drove the fruit bats to extinction. 10 or 20 years after World War II, okay, you see this huge increase, a 100-fold increase in the incidence of neurodegenerative diseases among the native Chamorro people. Not the Americans or Asians who were living on the island of Guam. They were not eating fruit bats, just the native Chamorro people. Okay? And then 10 or 20 years after the bats go extinct, you see the incidence of the neurodegenerative diseases go back down again. So there's like a 10 or 20 year delay between when they're eating the fruit bats and when you see these diseases develop. Okay? And then a colleague of mine, Paul Colley, he was an ethnobotanist taking a very different approach to trying to understand this, because the medical community for many decades, they couldn't figure out what the linkage was here. And what Paul Cox found out was that he calls them flying foxes, but they're technically they're fruit bats. They eat the seeds of the cycad trees on the island, and those cycads have symbiotic cyanobacteria living on their superficial roots. So these cyanobacteria producing this BMAA compound, and then it biomagnifies up this unusual food chain to the fruit bats. And so these people eating fruit bats were getting this huge doses of this BMA. Remember that number, 3,500. Okay? And basically in some of these villages in Guam, 25% of the people died from these neurodegenerative diseases. Okay, yeah, I don't know, you can't see the numbers, but basically, so here you're looking at BMA in the brains of these Chamorro people who died of these neurodegenerative diseases here. And these are your controls. These are Canadians who died of something else. No BMA in their brains, only the Chamorro people. But here's 2 Canadians who died of Alzheimer's. And presumably these Canadians were not eating fruit bats from Guam. So there's got to be another pathway to get BMAA. So a colleague of mine at the University of Miami Medical School, she runs the brain bank there. So she takes a bunch of Americans who donated their brains to science, and sure enough, all these people who died of ALS or Alzheimer's have high levels of BMA in their brains. Americans who died of something else, you do not find BMA in their brain. But you can now still argue, what's cause and effect here? I mean, you could argue that if you've got a neurodegenerative disease that causes your brain to accumulate BMA, well, you can test that by using Huntington's disease. We know that's genetic. They've got the neurodegenerative disease, but you don't see the BMA. So that basically tells you the causation pattern is that BMA is leading to the neurodegenerative disease rather than the opposite. Let's go back to Florida Bay. Little cyanobacteria, you get a couple of shrimp from that location. Here's a shrimp, 2,000, well, like 3,000, almost as high. That's the amount of BMA in that shrimp, almost as high as the fruit bats of Guam. Another 1,500, half that of Guam. I did talk about this bloom. This is a bloom that occurred in Florida Bay, and in South Biscayne Bay. And I didn't have really good samples. I had to get some from a colleague after the end of the bloom, and it was up in this area, so it's not ideal. So there's a lot of variability in the data and so on. But here's a blue crab, 7,000, twice as high as the fruit bats of Guam. There's another one, 5,000. Here's a pufferfish, 7,000. Clusatch River, get these blooms of cyanobacteria. Here's some fish. ranging from 500 to 2,000 or so, 2,500. Okay. We know we get blooms of cyanobacteria coming out of the lake, going down the St. Lucie Canal into the southern end of the Indian River Lagoon. And the Indian River Lagoon is also notorious for getting blooms of cyanobacteria. So I wanted to go to the very top of the food chain there. So I got a permit to get brain tissue from dolphins that had died in the Indian River Lagoon. Okay, it's an endangered species, so you have to get a permit and all this. But the good thing is, when they find a dead dolphin, because it's an endangered species, they do a full autopsy, find out what killed it. It just shows the blooms in the St. Lucie estuary there. Okay, so we got tissue from 6 dolphins. This dolphin here, no BMA in its brain, but the autopsy showed it had been hit by a boat. The other 5 all have high levels of BMA in their brains, similar to that of people who died of say the Chamorro people or the Americans who died of Alzheimer's and so on. And the autopsy could not find any obvious cause of death. So it's sort of suggestive. And I've talked to at least 2 different people who study the behavior of the dolphins in the Indian River Lagoon, and they've told me they've seen dolphins that seem to be getting lost, just sort of wandering around, unusual behavior, sometimes swimming up rivers, up into freshwater lakes and stuff. Sounds just like an Alzheimer's patient. Okay, quickly, the mechanism, the bacteria introductory biology real quick. Well, it turns out BMAA looks very similar to the amino acid serine. You need serine in all your proteins. So what happens is, in translation, as you translate your DNA into protein structures, the transfer RNA is supposed to pick up the serine and put it into the protein as you synthesize a protein. So a normal protein, we can just envision like this slinky here. Okay? But what happens if you put BMAA in the place of serine? That changes the 3-dimensional structure of the protein. Now it doesn't function properly. And it causes the protein to get all tangled up, like this slinky here, okay? Which is characteristic of neurodegenerative diseases. You get those protein tangles slowly accumulating in your brain over the course of 10 to 20 years until eventually kills those brain cells. Okay, finish up here. The pharmaceutical industry looks for all kinds of unusual compounds produced by cyanobacteria, as well as, they go around the rainforest, coral reefs, everything, looking for chemicals that might have useful pharmaceutical properties, antibacterial compounds, antiviral, anti-cancer, and so on. And in doing so, they've discovered well over 1,000 different types of unusual chemical compounds produced by cyanobacteria, with all kind of complicated structures here, okay? The vast majority of these, we have no idea what they may do to us. But if you're exposed to these blooms of cyanobacteria, you're getting exposed to those compounds. So let's go back to this thing here. Some of those compounds, we know they smell bad. Some of them make the water taste bad and so on, okay? Those you detect right away, you know you're exposed to it. But what happens if some of these compounds, which we have not, it's hard to do research on something that only has an effect on you 10 or 20 years later. We discovered BMA totally by accident. How many of these other thousands of compounds might lead to long-term liver damage, neurodegenerative diseases, or cancer? We have no idea. But the bottom line is, you are being exposed to those compounds if you're exposed to these blooms of cyanobacteria here in South Florida. So what I'll do is I'll leave you with a quote by a famous philosopher of weapons of mass destruction, and I'll let you read it on your own, and then you can ask questions. Any questions? Yes. Well, I mean, ignore the politics and just look at the science. The simple, well, I do very bad at politics. from my point of view, number one, stop the sugar subsidy. Those farms are not economically viable without that subsidy. Number two, turn off the pumps. And they're using our tax dollars to constantly pump the water out to keep the sugarcane farms dry. So just turn off the pumps, let them flood, and it'll turn it naturally back into a wetland. Now you go from being a nutrient source, that Everglades agricultural area, it goes from a nutrient source back to being a nutrient sink. And that cuts off the flow of nutrients to all these water bodies where you get these blooms of cyanobacteria and red tide. But as you know, sugarcane farmers pay a lot of money to the politicians, and we've never been able to get anywhere with that.